Welcome back. I almost feel like a show leader. So a show leader would start a session like this. Do you have a good time? Do you like it? Do you want more? Well, everyone seems to be smiling, so I take that as a good sign. We have another 2 hours to go, a busy schedule. I cannot guarantee that I get through all the slides, but let's the most important parts at least. And what I will start with now is first ask you any questions that have emerged during lunch. Now is the time to ask before we hit the next topic. Yes? Yes? Please? Yes, well, the pilot scale, what you do is you get from the supplier these bigger cans, and then you push. You cannot pour them as you do in the lab, so you have to push them with nitrogen gas into the reactor, or suck it into the reactor even by suction. So handling large quantities of dangerous chemicals poses technical problems, technical challenges, but we can deal with most, and if we cannot deal with them, then there are always expert companies out there who have the technology handling phosgene, for example. Who would dare to do that unless you could poison the whole community? So the butyl lithium we did like that, and other times you have to have concealed charging if it's a solid support. Some substances we are handling are carcinogenic, and you need to be, you cannot spread the dust or anything, so you need to have it concealed in a hood maybe even, and charge it into the reactor. So it's a good question, and you have to watch out, especially when you go up in scale. In the hood, in the laboratory, you have the glove boxes, and you can manipulate small quantities, but when you talk about kilograms and liters, it's a different ball game. Sure.
So we have a couple of compounds that we call the use of lithium, and it's made by a company, a German company called Chemical, and it comes to the factory by train. So wagons are filled.
Yeah.
Here is butyl lithium diluted, and then it's pushed by nitrogen.
Yeah.
You can imagine trains coming.
Yeah. Yeah, I know. So, good. Let's have a look at this molecule. An antidepressant, 5-HT1A agonist. There are many subclasses of receptors. HT stands for hydroxytryptamine or serotonin, and that's what I talked about in my first project, Simaldine. The compound that I forgot to mention, which made it to the market and was a really big success, was obviously, which was later than Simaldine in development phase, but they didn't have these problems that I mentioned to you, so they were successful where we weren't. This is one of the rare cases where we couldn't find a salt. It was just impossible to make a salt of this, and that has to do with the congested environment around this nitrogen. If you build a model of this compound, you will see that it's very difficult to put in a counter ion there and make a salt of it. So we had to use the free base. This went into healthy volunteers, but it had to be stopped there because of side effects. So it never actually was checked in depressed people. There we go. A long and tedious linear synthesis. This has been published, so I won't go through the details. It's a relatively simple starting material, resorcinol monomethyl ether. You protect the free hydroxyl group in butyl lithium. You formulate using DMF as the CHO equivalent, pretty standard textbook chemistry. You use a Michael addition onto acrylonitrile that brings in this 3-carbon fragment. And then it doesn't stop there, because the anion that you generate here will couple with the formyl group here. So you go from this aldehyde to this chromane skeleton. These are called chromanes in one step, more or less. Hydrolysis, nothing much to talk about. Saturation of the double bond here. And then go from the acid to the amine by taking out the carboxyl group. It's a name reaction, as you all know, the Curtius reaction. Instead of using hydrozoic acid, which is not very pleasant, you use this reagent, diphosphoryl azide, very expensive, and you just use one nitrogen equivalent here, which, as you can see, this is the nitrogen. So I think of the molar mass of this reagent, you just use 7% or something, very small. So it's not atom economic, as you can imagine, but it works. You capture it as this carbamate, and then you hydrolyze off this group and the benzyl group and the carboxyl group, and there you are, closer to the target now. But it doesn't stop here. You have an end game to deal with. This was a racemate, as you noticed. So you have to separate the enantiomers. You do this by classical diastereomer resolution using tartaric acid. Worked pretty fine. I'll show you some data about that. Then you have to attach the 2 alkyl groups. And funny enough, if you do it in the reverse order, you start with the then try to attach the isopropyl. That doesn't work because of the congestion. So you have to do it this way. First, the big group here, and then this one, which is smaller, then it does work. But it's a tough call to attach these two things. If you want to add bigger groups, you will not succeed. So there was a whole series of tertiary amines evaluated around this theme, but this is what finally came out. What remains now is to go from the methoxy group, boron tribromide treatment, to the hydroxy, activation with triflic acid here, and then look here, carbon monoxide, enough to poison the whole village actually in the last step. And this is a fantastic reaction, palladium catalyzed obviously with a diphosphine ligand here, palladium 0, isoprene. So you capture the acyl intermediate going directly to the amide here in pretty decent yield. Again, running this on the pilot plant, you need to know what safety precautions to take, because if it starts to leak out carbon monoxide, people are in a place you don't want to be. So, but we managed this. And this is the focus on the very final step, which is probably the high technology end of it. You have to be very cautious about the relative pressure of carbon monoxide here. You see, it's a small interval that you need to hit. What happens if you go higher? What is the main side reaction in this organometallic transformation, in this carboxamidation? It's actually introducing a second carbonyl group. And that is, we saw it also, if we stick to this tight interval, we noticed there was a dicarbonyl there, so you can't really be 100% selective, but it was relatively little. DMA as a base here, quite good yield. We were happy with that. The whole sequence is very long. It's linear. You have to dare dealing with carbon monoxide on scale. Palladium, again, in the final step, it's a very tight regulatory level that you need to be under 10 ppm. And the process solution that gets out in this reaction has about 1000 ppm or something. So you need to come up with a way to purify to reach 10 ppm. We were always successful with the filtration and extraction procedure to get this out. So, and you can add scavengers that attach heavy metals to them. So there is quite good technology available to reach these levels. We knew that we had to preform the active catalyst. That's step one in this scheme. I already mentioned the relative carbon monoxide pressure. You have to keep meticulous control of it. Otherwise, you would see the biscarbonylation. And then the sophisticated extractive workup. We ran this chemistry at the 40 kilogram scale. I think pretty impressive. So we would have liked to go on with this project, but these are the publications. The first one goes back a number of years. So unfortunately, this had to be discontinued. The overall yield from the first pilot plant run was not more than this. So people were they worked on an antidepressant, but the workforce, the team came out to be very depressed themselves because the target, it's bizarre, the target was to make half or 1 kilo, and we didn't succeed. All sorts of problems occurred along the way. We started with pretty large amounts of the phenol, the starting material, and 80 gram came out. So this was far below expectation, but the purity from an enantiomeric point of view was excellent. We couldn't hardly detect the wrong or the antipode. The formulation went much, much lower than we had expected. We did have no control over the formulation, yet it's described as a very trivial step, but if you go up in scale and you don't know what you're doing, this is what you will face. And the carbonylation, because of the purification issues we had no good method here, also came out as, and this is devastating because it's the final step. So you see we have a fair amount of material going into the last step and then just more or less nothing comes out. That is what causes depressions amongst chemists, I can tell you. The diastereomer crystallization wasn't robust. We had to chromatography in the form of a silicon dioxide filtration, and it took us an enormous, can you imagine 3 to 5 people working with only this sequence almost half a year, and you get out 80 grams. This was darn expensive, I can tell you, but the project had some material to work with. So what do you do about that? You tackle the weak spots in the process. You optimize the way that butyl lithium, and this is a known situation, how you add it to a reactor above or below the surface, and a few other fixes. It raised the yield quite dramatically, almost doubled it. The same chemistry, but with a better procedure, I would say. We put the diastereomer resolution, a much better yield here. We replaced this nasty reductant to this much better controlled one. Hydrogen with BR3 is a nasty liquid, heavy, fuming. For those of you who have used it, I don't recommend it. In the lab scale, yes, but not on the pilot plant. So we could actually change it. It's quite interesting. The same chemical transformation can be achieved in this system as well as in refluxing HBr. You wouldn't really immediately believe this is true, but it is. So the opportunities are there. Flash chromatography for purification instead of a tedious silica procedure, and then we trim the removal in the final step. This is just to demonstrate what you can do with diastereomer resolution. This is the procedure when we started. Slow cooling, textbook often says you have better control of the crystallization. Only what you want to crystallizes and not the rest. Well, that didn't turn out correct. The crystals were not that pure, 85-15, so you lose a lot there. So some smart people tried to optimize it, and it instead was a rapid cooling that was the clue. It's quite opposite to what you learn in school. Go slow, but here you should go rapidly, and then you need to know what the trigger temperatures are and where the crystallization starts and how soon you need to isolate, because if you wait too long here, the wrong antipode starts to crystallize as well. So here people need to be on their toes, reaching the temperature, start filtration. So again, it shows you need to be close to the chemistry. You need to observe. That's my recommendation. The purity was brilliant here compared to the previous procedure. Now we go to the next molecule, and this is a relative to the previous one. You recognize the chromine skeleton. You recognize the amine substituent. You have different groups on it, and here you have an amide instead of what we had in the... Yeah, it was an amide there as well. Sorry, I forgot. So it's an amide in both cases, but the key differentiator is the fluorine up here, which you didn't have in the previous one. So it's also working on the serotonin receptor as an antagonist. Here a salt could be made, a tartrate, increased neuronal transmission. That's the current theory of how you can alleviate depressions. You have to maintain a high active concentration of serotonin in the gap between the neuronal ends. So, and if you block the reuptake, then the depression might go down, and this is one approach to it. This one actually went into patients, phase 2, but didn't show the efficacy you wanted. Again, a dead end, but it gave us some nice chemistry. Every decent person having the previous project in mind would have started their considerations with this, or even here, the desired enantiomer of the chromine, aminochromine. So much is already in place here. You think, I'm almost home. This is what medicinal chemistry gave to us, and we believed, yes, that's good. We have access from the previous project to large amounts of this chiral amine. You have to brominate here. You introduce the cyclobutyl groups early on. As it turns out, the most costly components in this whole synthesis is cyclobutanone. Believe it or not, a very trivial compound, but it's difficult to make. It's a Mervine ring expansion of cyclopropyl methyl alcohol. acidic conditions. So, but this could be made. Then comes the tricky step, the fluorination. And this is really for experts in fluorinations. So you have a exchange with lithium here at the bromine position. That's relatively straightforward. Then you have a fluorination agent where fluorine appears as F+. So this is a diamide N-fluoro compound. So again, of this reagent, you just use less than 10% of the molecular mass. So it's an expensive way to get your hands on the fluorine. Very low temperature, and it requires chromatography. We knew this. We had tested this in the laboratory. But we felt confident we can fix it. One of the here is that this kind of chemistry always introduces hydrogen here. There is always some humidity left, etc., some water, as we spoke about earlier. It turned out, once the pharmacologists got to know these compounds better, they noticed if this is an antagonist, the hydrogen analog is an agonist with 10 times the potency of the fluorine compound. So it counteracted the fluorine. So we were given a level at 1% of the hydrogen analog to meet in the specification, and that was tough, as I will tell you shortly. So after the fluorination, you have to spend several steps to manipulate this hydroxy group into the carboxamide. Similar chemistry to in the previous project. Triflate, ester, here you go via the ester, the acid chloride, and then you, so it's a more sluggish type of procedure compared to in Balsotan, and then you prepare the final salt. So what happened? We have published on this quite heavily, so it is out in the domain. If you are not running under very strict conditions here, the lithiation will work, but if you have not control of the amount of protons available in the system by water or residues of solvents that are protic, etc., you will get a very high amount of the hydrogen analog, and that's what came out for us. About 30% of the process stream when we ran it in the pilot for the first time gave the desired product, and about the same amount, 35%, was the hydrogen analog. And then we had a starting material left. It was a mess. And you will remember that hydrogen and fluorine are very similar in size. So separating a hydrogen analog from a fluoride is extremely difficult. So what did we have to do? We had to run chromatography, and a very sophisticated one. And it was so sophisticated that we couldn't do it in-house. So what we did, we contacted a French chromatography specialist at the time. This was around 95, 97 maybe. So we shipped down all material, and they worked for 3 solid weeks on large-scale preparative chromatography to take out the fluorine analog and have the hydrogen analog discarded. It was a very expensive experience and very much work intense as well. So we realized that this is not a sustainable synthetic route. We have to come up with something else. In spite being in the golden positions to start with a very advanced intermediate, this is what you immediately feel, this must be a piece of cake. But we figured out that we will never be able to run this chemistry. We will never be able to control it. So, a new route. And this is a tougher question than the one I posed earlier on. How would you make this molecule in a different way? It will take you some time to figure out, I guess, so I will not give you much time to think about it. But it takes you in a quite unexpected direction. For us, the driving force was we needed to find a starting material where the fluorine was already in place. We didn't want to touch any fluorinations whatsoever anymore. It ends there. And there are a lot of fluorine-containing building blocks out on the market at fairly cheap prices, actually. So what would be a suitable fluorinated starting material? How far would you have to integrate your synthesis to meet that criterion? And also taking, that was the second criterion, a strategy where you would not introduce the cyclobutanone in a very early stage, but rather towards the end. It is expensive. We had it tailor-made for us by an English company, and it cost quite a lot of money. So it's better to introduce it towards the end and then push the price down. Well, I won't keep you in waiting much longer. Oops. Oops, where is my new route? Just a second. Did I miss on this? Did one slide disappear, or I'm sure it was there when I downloaded it. Improved route. Here should be it's empty. Frustration. It's empty also on the computer. I'll have to tell you quantitatively then. Oops. So we had to start with benzoic acid. So, shit. So we have the fluorine positioned here, we have the carboxyl group here, and we have the phenol here. So a 3-substituted benzoic acid was our starting material. And you can see the origins here. This is the first step, or one of the first steps in the sequence. This is the starting material and this oxygen. So now you get it. Sorry about this. The first chemistry you do is to attach this propargyl group here, relatively straightforward. But then comes the trick. How do you form the chromin? Yes, you use Claisen rearrangement. And this is a very interesting sigmatropic reaction, but it requires very high temperatures. And you will get a mixture of both 6-membered ring, which we wanted, and the 5-membered ring. And when we ran this first time on the pilot plant, we didn't want to charge all that we had of this intermediate, but we selected it or divided it into a couple of smaller batches, and I'm lucky we did so, because we always saw the furan come up at a scarily high level compared to our laboratory investigations, and to the end, 80% of the wrong isomer, 80%. What was happening? Well, we found out because this was made under basic conditions. So big scale, you will have your intermediates in big tanks, and then you suck out the solvent or the solution for the next step, and this we did. So the supernatant that you used for these earlier batches didn't contain any potassium carbonate. The final batch, we just suck it dry. We just took out everything, including all the potassium carbonate, which then destroyed the chemistry. It's a very hard way of finding out that you shouldn't do this, but it's good we divided it into smaller batches. So we established this through laboratory experiments, so we could nail down the reason for it. High volumes during workup, and this is for those who are interested in the mechanism for forming both the 6-member desired product and the 5-membered. Why do you get this? Well, this is the current mechanistic theory. You break this bond, so it's 3 carbons away in both cases. That's the reason it's called a 3-3 sigmatropic shift. You see the temperature well over 200 degrees. Again, very challenging. Very few pilot plants can run chemistry at above 200 degrees. So you have to go to companies that have the technology, that have the heating capabilities. Normally, a standard pilot plant would stop at 150, 160 degrees. So we liaised with an Italian supplier who could do this chemistry for us. So you break up this bond, you get this allynic intermediate here. The bond builds up between these positions. Then you can re-aromatize the 6-membered ring to get you the phenol, and then it could go in 2 directions, either via the carbonyl, and then a 2+4 cycloaddition gives you the desired chromane, or you could generate the phenoxy intermediate, which then ring closes on the 2nd carbon in this 3-carbon chain and gives you the furan. And these are test runs with different solvent. You need to have a solvent that really allows heating up to 200 and more degrees. And this is the composition of desired 6-membered ring, and this also is dependent on which kind of X substituent you're carrying here on the molecule. We have this one in our case, and you see with base the yield is close to 0. So we really proved that base is a no-go here. If you use ethylene glycol, most of the product is the 5-membered ring. We decided to go for diphenyl ether and stop the temperatures at 195 degrees. And this gave us a decent control of this ring-forming reaction. We had never thought that we would have to go through this complicated chemistry when we started this project, for obvious reasons. This was far beyond our imagination. Our spectacular improvement that we did to existing methodology in the literature from Hassner from the late 60s. And this is the olefinic nitration of the chromane. This is also one of the earlier stages. What he used in his paper here is sodium nitrite and iodine. It's thought that this is a radical process. I haven't seen anything of the contrary. And you will form nitrosyl iodide as a very reactive intermediate, and it regioselectively adds like this across the olefinic. This is not a benzylic or aromatic nitration. This is an olefinic nitration, which is a bit rare as well. The downside of this procedure, here on paper it looks pretty fine, but you need a massive excess of iodine. We would have consumed not the world production of iodine, but quite an amount, 4 kilograms per kilogram of starting material. I think on a molar basis it's about 3:1, so a really massive amount. Low yield, stability problems in this system, volume efficiency was poor, a lot of inorganic salts, as you can imagine, and complex workup. But when we tried it in the pilot plant, 45% yield. And I think the literature said 50 or 55 or something. So we were quite happy, and we had tested it in the lab, and we were a bit higher, but we were happy with this. But if you look at the chemistry, what's going on here, iodine, nitro group, this is what you want, but the iodine comes off again as HI. Isn't this resembling a catalytic opportunity? The iodide is reformed, so couldn't you do something here? Indeed you could. So after some thinking and experimenting, this is what came out. This is the company that we worked with in Milan, Zambon, very skilled company in this kind of chemistry, nitrations, high temperature. So the system is the same, so sodium nitrate, iodine. But as the reoxidant, we have Oxystrong, which is a brand name for a. So this is not a typo. This is per-acetic acid mixture with hydrogen peroxide with some water and acetic acid. You can reduce the iodine to really catalytic amounts because you reoxidize continuously. It's a cycle. You charge this mixture of reoxidant slowly, and the yield goes up to 70-75%. So we have a patent on this. We have published all the work around this process in a book chapter. I think it's beautiful. It's, again, sad that we couldn't commercialize this. So we saw a catalytic opportunity here. So the achievements in these projects, for about Sotan, the learning was that we started too late, and we kind of dug ourselves down into the medchem route and never managed to escape. We were stuck with the medicinal chemistry route, which we had to regret many times. But we were successful to scale it up to a decent pilot scale, 400 to 600 liters. We could produce 100 kilogram batches of the racemate, and this gave us about 27 kilogram of the final product. And the overall yield, which was 0.25, raised up to 3.7%. Maybe 3.7 isn't something that impresses you, but it's quite a substantial increase for this long route. But still, we would have had to live with a very expensive product, given that the optical purity was excellent. And a similar situation for Ribalzotan. We started too late with work on a new route. We were again stuck with something that we imagined. We locked ourselves mentally into having this starting material. It has to go. Prove it. Fix it. But we didn't manage. It was in vain. 15% yield, which was good, and we could recirculate the wrong enantiomer. Excellent stereochemical purity, process optimization, innovation, and 25 kilogram batches. And the cyclobutyl groups were introduced in the very last step. I can mention that as well. This is yet another neuroscience product, again in the depression and anxiety field. Builds on roughly the same type of motif, but it's not the chromin, it's a tetrahydronaphthalene and or a tetraline, the same type of receptor, but another sub-receptor than for the other projects. Discontinued in phase 2. Lack of efficacy. You've heard it repeatedly. The step from model studies in the neuroscience area to efficacy in man is very long. So I think neuroscience as a therapeutic area has one of the poorest outcomes in the pharma industry because of this gap between your understanding the biology and how well the compounds actually can treat the disease you're carrying. This, I just show you the third generation. There was a medicinal chemistry route, not a very nice one, as a first generation. The second generation used benzylamine here, but it was realized that, hey, we can have a chiral induction by using a chirolamine instead here for making the imine and then hydrogenate this double bond. It gives you not good of an outcome, but it gives you a decent diastereomeric excess. So this is the route that we pursued. We pioneered the Buchwald-Hartwig chemistry. This is in the early 2000. This chemistry had been out in the literature for about, well, less than 10 years. Easily, less than 10 years. And this is a tricky reaction. We obviously followed the Buchwald-Hartwig as much as we could, but we realized we need to do some changes. We found decomposition of the catalyst complex is a palladium catalyzed. You have BINAP. You could use, because this is not a chiral reaction, you can use the racemate of BINAP. You can use either enantiomer. As it turned out, one enantiomer was cheaper than the racemate, so we used an optically pure BINAP, but that doesn't have any effect on the chemistry. You have to have a base present, solvent. It's always a risk in these reactions that you debrominate, so you have to really monitor the amounts of reagents here, because otherwise you might get the H analog, which is a bit difficult to get rid of. The papers of Buchwald mainly, they always apply about 3 to 5% of palladium. We managed to go down below, it's a bit high, but in academia, people go for the safe bet. If I use enough of catalyst, I'm sure it will work. In industry, you cannot do this. You need to show how far down you can go, and we could actually go below 1% here. And we could even drive this further down on lab scale before the project was discontinued. And you have to optimize the BINAP amount and the base for the coupling. It's a fairly harsh reaction, 4 hours at 100 degrees, but it's quantitative. We scaled this up to a very large scale given the early stage of this project, and it went beautifully. 100% conversion, and we could keep control of the debrominated impurity as well. Had a nice workup process, so these are the final stages, maybe not so important. The group on the amine here, reductively precipitate of benzoate salt, and then the end game coupled with this big side chain here. There's a lot to say about this, but I'll skip that. So the total yield of this sequence, 7%, very pure material, high scale. This is what we made in total before closing the project. We tried biocatalytic approaches to make the R enantiomer, but we were not successful. There weren't enzymes available that could make this for us, a transamination on the ketone. Today this might be quite different, but in those days it wasn't. Choosing the right synthesis. This is a school book example of choosing the right opportunity of making a molecule. This is a class of molecules that we were working on. Tetralenes again. This is the medchem route. Just talk you through it. You start from this commercially available tetralone. You add cyanide in the form of TMS cyanide to the double bond here in the carbonyl. Very good yield. You hydrolyze it, a sluggish reaction, takes a long time. You have to be careful with the temperature because you get a lot of side reactions. Otherwise, then you hydrolyze it further down to something which we only could define as a complex mixture, which is something you would not like to see in your reaction schemes. But with chromatography, you can achieve miracles. You can take out all the garbage and you get out the pure product, albeit in very, very low yield, as you can see. But you can accept it, and especially in medicinal chemistry, they were happy. They had material they could pursue. You hydrolyze the ester. We introduced the amine functionality to get you to the amides, and then again chromatography, poor yield for a final step. This is what was existing when we started to work. 2% overall yield from the starting material. You have heard it before. What options did we see when this was presented to us? Scale up of the route. It worked. They had shown that it works. Some steps better than others, but it was a realistic opportunity. But at the end of the day, it was not seen as viable. This is probably not what we should do. There were a lot of issues, chromatography, finding enough of the chiral amine that we introduced towards the end. Modify the medchem route. Improve the nitrile hydrolysis, which was one of the bottlenecks in the beginning. Maybe because the molecule is chiral earlier on, maybe you could run the resolution much earlier, so you could avoid chromatography here, or even apply asymmetric synthesis in the cyanohydrine step. Again, this is about 15 years ago. There wasn't much available in this domain yet. There were a few reports where you could do asymmetric cyanohydrines, but it was seen as maybe a bit over the top given the timelines that we had. The final option that we went for, find something else. So you need to go back, check the literature, use your own imagination, your intellect and what have you, sit in a team, throw ideas at each other. This is what came up. Very smart. I think this is really elegant. The beauty is that it starts from the same starting material, but has a totally different strategy in how you make it. Here we see actually a Wittig reaction, which is tricky on scale, but the yields are impressive. Standard type Wittig, more or less. And then Sharpless chemistry with his sophisticated chiral inducers here. So it's a dihydroxylation. Very smart by the chemists who thought that this could give us the stereocenter we are looking for. It could give us a handle to go from the primary alcohol to the acid, which is a good reaction, and then just add the base. Brilliant. This is what I call innovation in synthesis. Nothing of this chemistry is new. It was all available. And this shows in a nutshell what a process R&D function is about. It's not about discovering novel reactivity or novel methods or synthetic procedures. It is to use, as far as you can, existing ones, trim them, optimize them, increase their performance. that in 9 cases out of 10, I'm sure this will solve your problem. And it did definitely here, because the most impressive part is, this is the yield. 2% in the previous case, I mean, this is quite a substantial improvement at an early stage of development. This, if you are in doubt, this shows you just how much time. Route A is the medchem route, route B is our route. You can see in spent time, less than half. How many man-month you have to spend, less than half. How much of this expensive and difficult to get at, a mean, do you need? And how much of the tetralone? It's a fraction of the other route, as you can expect. So this was a boost to both productivity, efficiency, environmental consideration. You don't need to process so much materials through this process. And we always try to motivate our chemists by saying that even if the project fails, everyone working in this area knows that they might never touch a successful project. So you need to keep them self-motivated, wanting to do the best, etc. Under every circumstance. You can always claim, okay, if it fails, but you can still say and be proud of the value that you have added. I think that's a good way of keeping people's momentum up. This is something which is interesting, which I think is important for academic chemists to know. How do we achieve stereoisomers in pharma? This is a slide maybe 10 years back. I doubt whether it's really different today. This is a publication by colleagues of mine in AstraZeneca and in Pfizer UK and I think also Merck UK, who looked through a large number of projects, 128, and checked published literature. How did the stereogenic center be generated? How did you get the chirality right? And this is what comes out. First, you can see that the number of stereo centers goes down very sharply with one. Yes, it's a fair portion. That has changed now. It was still 40% racemates in those days because it's a backlog of projects that you started maybe 10 years earlier when racemates were still more common use. Now you hardly see any racemates, even if it is not forbidden. You can develop racemic products. It's only that the regulators will demand from you that you document also the antipode. You have to demonstrate it. So you have to run tox studies and other studies to prove its, benign properties for the intended use. But if you raise the number of stereogenic centers, it's hardly that you find any compounds out there. This is maybe what is interesting. They checked where did the stereochemistry come from. Maybe not surprisingly, more than 50% of the cases, it's purchased. It doesn't mean that the problem doesn't exist, but you don't have the problem in-house. It's someone else who will do it. All these special companies, the suppliers of chiral building blocks, etc., they are hidden under this situation. So the preferable option, and I think this is still valid today, is to place the desired enantiomer or any enantiomer and then control the relative stereochemistry throughout the synthesis. Asymmetric synthesis that you in academia spend so much resource on, I alluded to this before, you don't find it too much. People are a bit afraid, it's complicated, not everyone is comfortable with developing an asymmetric synthesis, you have to handle transition metals, etc., etc. Interesting is that resolution had a very strong foothold here. Almost 1 in 3 projects relied on, and you saw that in my examples, relied on resolution, which is a stupid way of getting the right stereochemistry, but it's effective. It's just, once you find the resolving agent, it's just to define how you crystallize it, and you don't need to bother anymore. So I think this is quite an interesting aspect. It would be nice to see the situation today. Has there been any change, or is this still the prevailing approach? My answer is right now, I don't know. Enzymes. Still, there is an uneasiness among scientists, maybe more in development rather than in discovery. What are these beasts? What are these enzymes? Their origin, properties, structure? I really can't put my finger on them. I'm reluctant to use them on large scale. I've heard that many times. Is the predictable? Is the consistent? Conceptually, there is a problem. How do you understand? How does it operate? What's the mechanism? What affects the outcome of an enzymatic transformation? Time has moved on. It's going to the better, but still, in the generation that I belong to and others, they still have some difficulties in coping with enzymes. Yes, occasionally it's a very complex workup. If you run whole cell systems, it's a messy workup to product from a debris of cell material, proteins, all sorts of stuff. They're diluted solutions or mixtures, and how do you fish out your particular compound, etc., etc. The impact on quality, is there one? Can you predict it? And what would the regulators say? Do they agree to using enzymes for making active ingredients? Drivers. There are quite a number of drivers, and they are increasing in numbers. New enzymes have come to the playing field by various techniques. You can produce novel enzymes with novel reactivity, novel selectivity, etc., etc. You can find a supply of these enzymes. So it becomes more and more a part of the toolbox. People see them as realistic opportunities when they develop processes. This is very important. If you demonstrate via examples, small scale, large scale, whatever, people will be convinced. The laggards will be convinced that, hey, there is something more to it than just some nice reaction schemes. You can use it from a practical point of view. Green chemistry, I don't need to convince you that enzymes. So to convince someone who believes in green chemistry that biocatalysis is the right way forward is pretty easy. People are more open, show a higher degree of interest for using enzymes. And there are regulatory approvals with enzymatic steps, and I think that convinces at least the pharma industry that it is a decent way to proceed. Here we have 3 examples of commercial drugs where biocatalysis is an integral part in the process. One from AstraZeneca, this big seller, rosuvastatin. It's a lipid-lowering agent, cholesterol-lowering agent. This was once the world's largest single drug, Pfizer. Sorry that Simon isn't here, who could tell you the peak sales of Lipitor was about $13 billion. We will probably never reach that again. It was just so outstanding. our drug from AstraZeneca, 5-6 billion, so enormously successful drugs. And there were, at least I know it from this case, chemical routes to make this pretty complicated molecule. But what finally came out as the smartest, cheapest is that you use an aldolase, in this case, trimerization, if you wish, condensation to get this intermediate and then eventually to your final drug. And Pfizer chose another strategy for their cholesterol-lowering agent, two reductases in a cascade type of arrangement, gave them their compound. And not to forget about the Merck pioneering work using transaminases on this prochiral ketone. Together with Codexis, they found the right, because using available transaminases, the outcome was very poor, very little EE. So they had to develop novel enzymes to really achieve the selectivity that they needed. And this is the way that sitagliptin, a $5 or $7 billion drug, is produced today. With this key, there are chemical steps, and then there is this step at the end. So much do I have now? It's 1 hour more. Okay. Any questions about this section? I know it's fact dense, so you can take some time to digest that. If you have any reflections or any questions, please.
Sorry, going deeper in the effect or lack of efficiency of the drugs that you.
The neuroscience drugs.
Do you think that could be related to polymorphism, just about this reality? Because in the past, not so that act in the
In different ways, yeah.
pharmacological properties.
Mm.
Do you think to reanalyze the data, to pose or to test the clinical trials?
I mean, these all come from the past 20 years. So the knowledge about polymorphism was quite advanced already at that. We had cases in other companies where they had chosen the wrong polymorph and there were consequences. So I think more or less everyone, at least in their minds, were aware that this is something you need to watch out for. So I don't think that actually killed them. They worked to some extent as intended antidepressants, but they were not enough efficacious, or they had side effects. I know from the patients that were treated, because you give them the dose for a very short period of time to be on the safe side. So you cannot give continuously as you would give in a real treatment situation. These people started to have, or at least a few of the individuals involved, they started to have nightmares when the drug was taken off them. And this is some kind of adaptation which gives a bad signal, because how do you control this? Will it be used as a narcotic rather than as a treatment for depression? So there are so many negative factors that can change the direction of such a project, and which eventually might kill it. It might have secondary pharmacology effects like high blood pressure, constipation, sleeplessness, all these kind of factors, and you need to decide whether we can handle them and if they are acceptable. If it's a deadly disease and there is nothing else, if we had something for Alzheimer's with these kind of side effects, maybe the regulators would still accept them because we have nothing to offer, or for stroke or something. But where there is some treatment already on the market, it would be stupid to accept things that are not entirely clean. And that's the reason that safety and efficacy is so dominant in the reasons for why you discontinue your projects. So I think polymorphism, not that I've heard of.
Not could be related to side effects, but for example, side effects.
The problem is that our understanding of the brain, which is supposedly the most complex organ in the universe, some people say. That is certainly true for some people, I'm sure about that. We don't understand how it operates. There are models for where you have your different centers. I don't know exactly how realistic those are. How does the communication go? How do we learn things? How do we remember things? We know that. And then trying to interfere with a system that has its origin in the brain, it shows you the hurdle. It shows you how difficult it really is. That something affects the brain, we know that very well. Just have a couple of caipirinhas, we feel it. But in more, let's say, complicated situations, we don't have the answer. So once brain research really makes the breakthrough that we need to understand and can describe the biology of the brain, we are in a different position. Today we still have to feel our way. Okay, anything else? Reflections? Questions?
The ways that you get chemistry. Mention about the and we observed like in the last 20 years the revolution about the biocatalysis, and also the reductions, oxidation, electron sharpness. So I have these good tools. I really don't understand why you have just like you mentioned about 10% asymmetrics. What is the point? Doesn't compensate to investigate these reactions? Is it cheaper to buy the optical active compounds? I don't understand how in the pharma you don't integrate, this chemistry.
Cost. I mean, making new molecules is cheap. Chemists are cheap. They are not overpaid. The cost for developing a new drug escalates when it goes into the clinic. Then it starts to cost tens and even hundreds of millions of dollars. So here in the beginning, buying something which is expensive doesn't count as long as you can do it quickly. Here the time, the speed. If you have an idea and you want to exploit it, you want to be first, because only when you're first you will be able to take a patent. So you need to rush things, and buying something that a third party has done for you is much better than, developing an asymmetric, the self-oxidation. We worked our butts off for 3-4 years. You can't afford this in discovery in a competitive area. So that's the reason I think you see this kind of picture emerging. You pay what someone asks you to pay to deliver something you really want. And as you know, if you want something really intensely, you're willing to sacrifice something. In this instance, money. And money is not, in the early stages, it doesn't really matter if you do it yourself. If it's successful, yes, you can always try to find something else, but in the beginning, buy as much as you can. That's also behind the outsourcing strategy. Outsourcing is not for free. Someone has to pay the bill. But it's seen as a better way forward. You don't need to have all your chemists. You have like a factory of chemists nothing else than work 100% of their time on your compounds, day in and day out. Yes?
On the same direction that Julian was talking, decision to the option of sitting intermediate when we are starting synthesis, and how you decide and not going down on the because of the COVID synthesizing these intermediates is because of the regulation that starts from these intermediates, or is just what is possible for the industry or is readily available for buying?
It's a mixture of all those, except GMP. I've never come across a situation where you start to worry about GMP, because you know that this project might only survive the next month, even no one is interested. So GMP you can leave out. I think if you recognize the chemistry, if it belongs to a class of molecules you know a bit about, maybe you have synthesized analogs, that is obviously easier for you to pick that thread up and try to build on the previous experience. Literature precedents. Excuse me. Our task is not to invent new reactions. I have to repeat that. We have to use literature reactions as far as possible and improve them, as we have done in many cases. Sometimes, occasionally, we might come up with something quite new, but that's not really the purpose.
Yeah, but for example, when you have some hazardous reactions running, and it's preferred to go for outsourcing or not handling at the facility.
Yes.
As some
Yes, yeah. As I showed you with the fluorination here, we did it once and it almost killed us. So we wouldn't have done it a second time. We would have gone somewhere else. Now we changed the synthetic strategy entirely, so we didn't have to worry about that. You have to realize that you cannot have all the expertise in one organization. I don't know how big that would have to be. So you need to rely on partnerships with people who know these things, who have the technology, the experience, who can do it for you. And there are so many out there in academia, but also in industrial companies that will offer their services at a high price, but that's no problem. Price is never a decision factor unless it's over the top. If someone tries to screw you and really ask for payments that are far out, and because we always check what is a decent cost for this material. So it's difficult for someone to really come up with an offer that is just no-go, not acceptable. So I think that is where we are.
Maybe I could add something.
Yeah.
On that. I mean, in Bayer, the rule is you buy the most advanced intermediate industrial scale. Minimize the number of steps. You buy them on the producers. We have 7 to 10 steps. So you need to get the most. Say it, you are specialized. You are specialists of certain type of chemistry. No need to reinvent the wheel. Fluorine, this kind of boron. Go to the guy that knows.
Flow chemistry
We
gives
have
it.
one problem one
Yeah.
day that we have one intermediate, one key intermediate in only one company. That is dangerous. And if the pilot plant from them burn or there's an accident, then you're stuck. So we try to have two, at least partners, taking the same intermediate.
Yeah, but when you don't have option,
You make it.
yeah.
You make it. There is no way out. I mean,
that's the weakness working with suppliers. We lose control. We are mindful of it, and we have to sacrifice, because if you do it in your own facilities, you will have access. You will see the reactions, you will have all the primary data, you will see what's happening. But if you let somebody else do it, all this knowledge is lost. Yes? So people need to, it's like having the cake and eat it. You can't do both. You have to decide, what do I want? What do I want? So this is a response to the question about dangerous chemistry. I think, and I will show you, hopefully I have time, a few examples that we have I think this gives an opportunity for chemical companies like the pharma industry to actually produce or run dangerous chemistry under very strict control with very good outcome. So this is a new opportunity. There are virtual companies, meaning there are just a few people in the organization and they buy everything. They have an idea of a compound. We think this could work. Maybe they have some biology data from a university lab, etc. But they virtually need to have partners for all the steps in the development to a final medicine. And the problem for these companies is that they can't evaluate the validity, the quality of the work that others are doing for them. They are, but they don't know. Develop a route for us, yes. Pay us $1 million. Oh, it's expensive, but okay, let's go for it. You can do that, but as a customer, you would like to know, has it been worthwhile? Did they do a good job? And if you don't have the competence to judge, you can easily pay for very inferior type of work, and this has happened more than once. Okay, should we press on? I will not spend much time. This is an unpublished case. This just shows you how far the medicinal chemists are prepared to go to complicate molecules. It was developed in Gothenburg as a cholesterol level lowering agent with a different mechanism than the Crestor's and the other types of drugs. So it actually stopped the transfer in the intestine across the ileal bile, or it stopped the transfer through the lining of the intestines by operating on the bile acid transfer. Smart idea. This is a 7-membered ring. It's a sulfone. It has a sulfur in it, very functionally dressed up molecule, large number of steps, and this is what it was. These are the steps. I will not dwell very long on this. I leave it to you. I have some text here to really enjoy how this was solved. We have the problem, we have the solution, we had to change strategies. The interesting thing I could comment on, here you have butyl groups, and why do you have 2 butyl groups here? It's to make it lipophilic, so that it can penetrate the lining of the intestines. So why all of a sudden do I have a double bond here? Doesn't that look very strange? It stays on with the double bonds. The double bonds are still there. They will be taken off at some stage. This is maybe the nicest chemistry, how to achieve the selectivity for the sulfoxide and not to get to the sulfone. So a lot of systems were tested, some obvious, others less obvious. What turned out to be the winner was this tungsten oxide derivative with hydrogen peroxide. High selectivity, good reactivity, but that's aside. Here, the double bonds, why are the double bonds there? You had to be cautious about the Heck relation by virtue of these double bonds. They could alkylate because this is a relation to the phenyl derivative here. With the iodobenzene. So anyone dare to make a suggestion? The final molecule is the saturated side chain. Why couldn't you put that on from the beginning? I have said it earlier, so this is a test if you remember what I said a couple of hours ago. It's a patent reason, because we found out that an angry competitor of ours, I won't mention who, had a patent blocking us because one of the intermediates in our long sequence had exactly that structure that they had patented. So we reasoned that we will never get a patent. They will block us. We had no reason to believe that they would give us a license. Why should they? Because we competed in the same area. So that's the reason that they had unwisely not patented the olefinic substituents, and that was our savior, so to speak. It's a fascinating project that unfortunately didn't go very long, but you can imagine to develop all this complicated chemistry that took us some time. Pressing on, environmental issues. This is again a review carried out across the pharma industry. If you analyze the PMI, the process mass intensity, which you define by summing up all the materials that go into the process by the number of kilograms of product you get out, that gives you the PMI, which is a more decisive factor compared to the E factor, which is what has appeared more in the literature, at least in the beginning when you started to calculate these things. So if you see how, in general, a PMI based on these 7 pharma companies is built up, it's a lot of solvents, 50% or so. These are indicational figures. Process water, 80%. Solvents and process water contribute to the PMI. The reagents, 10, maybe 15%, and then other aid chemicals, catalysts, filter aids, and stuff like that. So you need to be mindful of this. Also, a lot of the energy that is consumed, because if you do a real life cycle analysis, you also need to involve energy consumption and the origin of the energy if you want to do it really properly. Often energy is left out, but a large portion of the energy that you consume in a process for making an active ingredient goes back to the solvents. And a large portion, even if they are cheap, because you use so much of them, a large portion of the cost as well. If you look over the time scale, you often start with very high PMIs, several hundred, thousands even, and this indicates it's a complicated process and the sequence is not very well optimized. You have a lot of excess, you have diluted solutions, everything that you shouldn't have. But the trend is clear. You can take it down. And this is again a relative scale, but you can often, in a decently long, and the standard length of a API process is about 8 steps. You can go down below 100, maybe below 50. So there is hope. This is the same thing that we talked about. A lot of solvents build up the material spending in synthesis of active drugs. This is a benchmark study a couple of years ago, again 10 or 15 pharma companies. So it's statistically fairly well underpinned. Water and solvents 80% or more, and then reactants and other chemicals needed. So this is the picture. Funny enough, and this wasn't actually planned, we heard a lot about sildenafil, or Viagra as it's called, yesterday, and it just is a brilliant connection with what I will talk about, and that's the achievement by Pfizer. Simon didn't mention this, if you remember, how they have trimmed their chemistry, what they have achieved, and it's really amazing. This is where they started. This is the amount of solvent. They had all these solvents, you can see here from this bar chart, 1,300 liter, 1.3 cubic meters per gram of the active ingredient. That's incredible. But that's where medicinal chemistry stood when process R&D took over. Well, they actually optimized it a bit self, and fairly quickly they managed to drive it down to 1/10 of this. And then the commercial route, I mean, you see the trend here, 22.7, and future target is actually the current process. 4 liters with considerably less number of solvents, and what is good, the red color has been chosen for a reason. It's a red flag for an ozone depleting, or at least solvent you should try to stay clear from, methylene chloride. It's very volatile, and it definitely contributes to contamination. So with active work, you can make a better process. That is the message here. And Peter Dunn and his team have done a fantastic job, and it's been recognized by the Presidential Medal in Environmental Chemistry. These are a couple of novel compounds. Again, I will not go through the details. This is from oncology, so this comes from the former Seneca organization. You work yourself backwards to decent building blocks from the target compound. You need to identify what is your main task. Here it's to come up with a synthesis of the pyridone. Medchem has a route. You can enjoy it. There are bottlenecks that are indicated here, quite a number of them. Finding the starting material, its chromatography, the yields can be debated, chromatography again, methylene chloride, it's this side chain which is not easy to get at. It's using this very strong base, and again the strong base. You could do better, but an intermediate was identified that could be a good. This is a new concept, a strategic intermediate as a potential regulatory starting material. Because when you document a process in the eyes of the authorities, you don't need to document from a GMP point of view the whole sequence. If it's a 10, 15 step sequence, if you can identify an intermediate like this, that is stable, it's well characterized, you can make it, etc., they would accept this as your starting material. So your GMP process starts here and not here, which releases the resources that you would have to spend in order to define all these earlier steps as GMP or GMP quality. So that's the reason you have this regulatory starting material concept, which is very good. So these are the modifications that process chemistry did. A bit of different strategy, better control of the formation of the pyridone, but you're still grappling with the base. Twice you have the side chain unchanged. And then a further optimization gave you this compound. This is where we started. How can you characterize it? It's expensive starting material. It's a long linear sequence. Many intermediates have to be isolated. Selectivity is a problem. A lot of chromatography. The side chain is hazardous when used. And then you have separation problems, and you have two low temperature steps, and then the supply of this lithium hexamethyl disilazide, which is quite nasty and not manufactured by very many companies. The new route, like this, shorter, high yielding stages, only 3 isolations, better selectivity, no cold chemistry, and good atom efficiency. If you demonstrate this to some decision maker, they will be convinced that yes, it's a good job. Without understanding the chemistry, they will, in relative terms, understand that this is better than the other one. And that's the message you want to drive home. This is better. So continue to give us money, and we will. This is what we will deliver to you. The cost goes down considerably from $5,000 a kilo, more than that, to a third. And you would, if you make 50 kilograms, you have saved $200,000. It's not bad. That will convince even a stubborn manager, I can tell you. This is another interesting project, quite esoteric molecules. And this is an intermediate that everyone homed in on, called Inca. This is the medchem route. As in all medchem routes, there are weaknesses, bottlenecks. This triggered the look for alternatives, low yields, nasty reagents, etc., etc. So, starting from Inca, which was identified as probably best starting material or building block for this kind of compound, all of a sudden, Diels Alder appears here. Remembering how the starting material, or the target compound looked like, would you immediately connect this to Diels Alder chemistry? I'm sure you wouldn't. No one did. But we had an aid. This one. It's a software that we have developed together with a German software company called Infochem. It's now commercial, so you can get your hands on it. This software tries to mimic how the chemist thinks. It's not easy, but I think they are on the way. There will be further developments of this software, but it's pretty user-friendly. This is not the type of expert systems that Corey and Todd Wipke, etc., these people started to work on in the 60s. I mean, retrosynthetic tools have been on the agenda for decades, but no one has really convincingly been able to produce something that the bench chemists can use. They are always expert systems that require high-skilled computer people, not synthetic chemists. And this was the aim, to do something for the working synthetic chemist. So this system identifies bonds, atoms that are changing through a synthetic sequence. What is changing and what is constant? And there are, of course, algorithms building up this quite complex software system. What this system looks for, contrary to, let's say, normal literature search, is unprecedented routes, something that is not available. So when we wrote a paper that I will shortly refer to, we tried to sell this as an inspirational tool for the chemist to think in a new direction. And I think chemists know a lot of chemistry and a lot of reactions, but maybe they need to be directed in certain areas. And there is no chemist born yet who knows all the reactions in the literature. I am convinced about that. So you need some mechanical tool that can support this. You can present the data, the findings of the retrosynthesis analysis in very nice ways. And this has now been out on trial with a couple of companies, and it's really been met with open arms. This is our paper, one of the most cited and read papers that I've been involved with, just published 4 years ago. A team from AstraZeneca and from Infochem. We describe both the mathematics and the programming behind it without giving away company secrets, and then we give examples side by side what the system can do compared to what the literature shows. A very powerful tool. So for all of you who are involved in making complex molecules, may be worthwhile. This is available. And this system brought us to the Diels-Alder reaction, something that no chemist, and we asked quite a number, did not identify. I wouldn't have done it either. So this is the, because you have to go back to this furan to formulate it. You bring in this amine part. You open up, or you're attached to the nitrogen. I think this is ingenious. This acyl compound, and then you have the 2+4 situation dressed up for a Diels-Alder. Perfect. In 75% yield without further optimization. I'm impressed by what the machine told us. Then there is always a backside, a downside. The Diels-Alder chemistry was so excellent. No problems either technically or chemically, but the simple, in inverted commas, furan chemistry on the top here was a mess. What you think is trivial, this conformation, it brought us a lot of headache. So, again, comparison of route. The message you've heard before, there are weaknesses in a medchem route, obviously, cryogenic stages, a lot of intermediates that have to be isolated, palladium chemistry towards the end, an intermediate that can be isolated. I mean, you don't want to see this. And this is the Diels-Alder route. Very short, 2 stages to isolate, or 2 stages to work, and 1 isolated intermediate. Cheap reagents, no cold chemistry. Perfect. The final example is this bizarre molecule, an anti-cancer agent, medchem route. I could emphasize that this reagent actually stands for this side chain, trichloroacetyl isocyanate, which you on which you add the amino functionality here. So this comes from this reagent. Yeah. As always, there is chromatography involved, steps that don't work very well. The yield from this quite expensive intermediate, 9%. The new root concept was conceived based partly on literature. So the question, this was known from the literature, so would it work with a more complex acyl thiol compound, or would there be problems? I wouldn't be standing here talking about it unless this worked. It did work. So this is the chemistry. %48 yield, 15 kilograms were made. Comparison of roots. The original root, low yield. A lot of excess of the expensive piperidine building block. A lot of bromine had to be used. Trimetal aluminium had to be used. Robustness issues, again, things you don't want to see in a process. A lot of solvent, 3 tons for 300 kilogram of starting material, and the new route, shorter, convergent, much better yield, no aluminium issues, much less solvent. And this is the cost implication. It saves $16,000 per kilogram going to this novel method. So these 3 final examples show that de novo synthesis of aromatic rings is something you will have to consider more and more because of the architecture of the molecules. They get more complex. You cannot just buy them off the shelf. You have to produce them yourselves. That is something that's happening. The development of novel processes brought more convergent routes, much more atom efficient ones, skip all these esoteric reagents. You can do multi-kilogram, which are ready for further scale up. And it also demonstrates the different regimes under which medicinal chemists and process chemists operate. They have different duties in that sense. Crystallization, I've already spoke about the importance of crystallization in the pharma industry. Robust crystallizations, it is a key step in ensuring the quality of your product, the production efficiency, estimation. 1/3 of the pharmaceutical molecules are capable of forming hydrates. The question is whether you want one or not. Process analytical technology is technology where you actually measure live processes before they have reached the final stage. And you can do this also in the crystallization field with a technology called focused beam reflectance measurement. Maybe some of you are familiar with that. This is the equipment. It builds on a laser beam that you irradiate through your reactors. You have to have a lot of control devices here in this setup. And you will try to measure a quasi geometric length to say if you have the right size, the right shape of your crystals. It's much about the shape, especially if you go for inhaled drugs that you would take directly into the lung. You cannot use crystals like this. They would stick to your lining. You have to have very rounded, micronized particles. So this is a very good tool to steer crystallization processes. Flow processes, this is something that goes for everyone working in R&D. Try to think in new ways, out of the box. It's a recommendation that holds in every situation. Don't follow what others have done before you without challenging it. Sometimes it's the right thing to do, but not always. The drivers for continuous manufacturing, speed. It's much quicker than scaling up a batch reaction. Bye, see you tomorrow. The safety aspects we have talked about. It's a hype around flow chemistry. You can hardly open a journal where there is no paper about flow chemistry. So I think it's an intrinsic push in the chemistry community that we have to be part of this. There's something in it. I certainly need to know more about it. And this has taken this area forward. Yes, it reduces the environmental impact. Examples are ample. Cost savings. You utilize your raw materials much better than running in batch. The blockers. What reactions can you run in flow? Can you run every reaction? Obviously not. There is still a challenge around reactions where you have deposits, where you have solid phases involved, oils that can clog the channels if the reactor is designed in a channel mode. So there is some insecurity about what reactions. Ian Baxendale, who is quite a high-profile flow chemist in the UK, he told me maybe 30% of all known reactions are amenable for flow. Maybe that has changed a bit to the better, but we are not talking about 80, 90%, the majority. We are still working with less than 50%, I think. Yes, a long sequence. How much of the sequence will be open for continuous manufacturing? If you have 10 steps, how attractive is it to have one step in a continuous mode? The rest you will have to proceed in the normal classical fashion. That's a question mark. Is it applicable also when making large molecules, proteins? And I'm not talking about automatic machines, I'm talking about real synthesis or antibodies. Is flow chemistry something you can use there? Regulatory concerns. Regulators always want to see batch documentation. And if something happens to a particular batch of material, you always must have this traceability to go back. When was it produced? By whom? Where are the raw materials? Where did they come from? What were the qualities, etc., etc.? How do you ensure this in a continuous process? There is a tool now called Quality by Design that I will show you a bit about what that is in a second. This is a conceptual approach that AstraZeneca has taken. The early stage, focus on speed. Late stage, it's more the economics that drives it. Does this give us any advantages? Here, the speed is the only thing, even if it's expensive. You can handle dangerous reagents, extremes of temperature, exothermic events. You have good control in a way because the reaction space is so limited. A good thing when you talk about larger scale is you make as much as you want, and then you just stop it. It's an easy turn-off valve, which is quite attractive. Scale out or scale out. Does everyone know what scale out is? Scale up you've heard of. You go from a small vessel to a big vessel, but scale out. You stay in a small vessel, but you repeat them. So instead of running in one vessel, you run in 10 in parallel. That is scale out, which is a very attractive concept cost, and a few other things. So this is the type of chemistry that has been exploited with success. Curtius amongst them. These are reactions that are with the largest cyclopropionation, Hoffman elimination, and then large-scale nitrations, obviously, which is always a key target for making under continuous mode dihydroxylation. This is a reaction that we did in this reactor, which we were part in developing together with Alfa Laval in Sweden. Art 37, small channels. You can stack up these reactor compartments, as many as you like. You just push your reaction solution through here depending on the reaction time you need, etc. It's brilliant. And you can more or less squeeze this into a hood. And you can produce, if you leave it on 24/7, kilograms of material. You don't need to worry. So the batch process for making this sulfoxamine, 30 to 60%, very variable. In the continuous mode, much better control. The reaction was fast, so it was very suitable, and the yield went up to 74%, much more predictably. This is a Grignard reaction also under flow conditions. Running Grignards on ketones is always, no, this was an ester, yes, on an ester is always tricky because you get a ketone as an intermediate, and the ketone can again add another equivalent of Grignard reaction. You can add triethylamine to capture, prevent the ketone to be formed in situ, and instead you protect the ketone in this way, so you get a fairly high amount of the desired product number 2 here, but some byproducts as well. But more importantly, you get a yield of 60% compared to 30 in the batch. Few words about the quality I spoke about. This is one of the major catastrophes that has hit the pharma industry back in 1937, admittedly a long time ago. It sounds similar, it's a very similar molecule. I don't know exactly how this came about, but this gave a strong signal. We cannot just rely on people's good judgment and knowledge. They need to be guided on how to handle this. So already after the war, first guideline for GMP was published, and this has been pioneered in the US. The FDA has been instrumental in this, so all credit to them. We have the thalidomide tragedy 60 years ago, the newborn children with the malformations. The important thing is that the molecule, thalidomide, actually bioracemizes, so even if you would have given what people think is the active enantiomer, it would have bioracemized because it has a labile proton. It would have created the wrong enantiomer, and this compound should have been killed, or should not even have been made, to be honest. So all bulk actives, what we have been talking about, active ingredients, they are governed by the legislation that the Food and Drug and the Cosmetics Act present. So by US law, you have to obey and comply with these rules. And as the US still has been and still is the major pharmaceutical market, unless you like it, well, don't bother about the US, but there is no pharma company who can survive without having access to the US market. So you have to follow these principles. So what GMP does is put the customer, i.e. the patient, in the center. It views what the pharma industry does with the eyes of the patient. we need to convince the patient that what we produce is done according to the best principles, the best practices, and at a consistently high quality. It covers the whole value chain from when the raw material is delivered until the finished product, the tablet. There are a number of principles underpinning GMP: the people, the premises, the processes, the products, the procedures, and the profit. It's very expensive to operate a GMP plant, I can tell you. That the price skyrockets actually compared to normal bulk production of chemicals. That's one of the reasons it's so expensive with drug compounds. And GMP is actually one integral component of quality assurance. In the early days, it was analytical control. People thought you can control quality. That's rubbish. You cannot control quality. You can control some analytical parameters, but that doesn't control the quality. Quality control is a much broader envelope where you look how was it done, did it follow the procedures, the descriptions, were there any deviations? So you don't only rely on the GC, but you also look what happened actually when this material was produced. It's opposite to analytical control, a much more detailed review of how you produce a particular compound. This came then about to generate GMP. This was starting to operate on a more grand scale in the early 1970s, 50 years ago. Quality assurance, then we go even one step further. If you control quality, the next stage is when you can assure, you can convince the user, the authorities, that you have control of it. You have the evidence that this is the quality I will deliver. And this is what still is the concept that we use, even if it's undergone some updates and changes. Quality by design is something that was started early this new century, and this is what it is. One thing that the farm industry has been accused of is slowness in implementing changes in processes. If you had a registered process, no one dared to go in and change it, because that would require authority approval again. You don't want to expose yourself to the authorities, and they would check you and maybe find stuff that they don't like, etc. But quality by design, the word by itself indicates what this is about. You build in quality in the design of your process, not afterwards. It's part of how you design a process. And this gives you an opportunity to develop reliable and robust processes that take charge of novel technologies. If you have defined your process in rather going from a particular point of operation to a space, then you can make changes. Maybe not all the changes you want, but you are free to redefine your process without going through regulatory approval, as long as it stays within the space that quality by design studies have proven. It opens up for continuous improvement. This is a big change in the pharmaceutical industry. You don't need to re-register. You can make continuous improvements. It could be small steps, maybe change to another catalyst, a better catalyst, change to another solvent, etc., etc., if you have documented it in your design work early on. This is how it works. It starts with the patient and the patient's safety and efficacy. You look at the quality attributes of the final formulation of the medicine. You define a couple of criteria and a design space, and that goes down and leads into the active ingredient, because the active ingredient is a part of the drug product, which is the formulated product. And here you do the same. You define a design space and a control strategy. This is a pretty novel concept, but it is about to make the pharmaceutical industry much more efficacious. We can skip manufacturability, I don't think so. This is more that you as a producer should ensure that the active ingredient you're manufacturing can really be made to the standards and the quality that you commit yourself to. So let's skip this. The green chemistry, you know these principles. That's not new to you. This is appears in the list. A lot of other things. How do you become green? Identify risks. Minimize steps. Catalysis. Don't use too much energy. Don't isolate. I'm repeating myself. I don't know which time I'm doing this, but process intensification, recycling, abatement, waste treatment. And these are again the figures of what you're actually about doing in the pharma industry. So many solvents in the waste streams, a lot of energy goes into handling of the solvents. The cost is high, the cost part for treating the solvents, buying them and treating them, and also the volatile organic components. This slide you've seen. Process metrics. This is a model reaction. You can see that in the slide pack when you get it, how you calculate process mass intensity, atom efficiency, carbon efficiency, reaction mass efficiency. Nothing novel. Everyone should know this pretty well from the top of the head. This doesn't add so much. These are a few reactions. This is now the end story. Which is the best reaction? We've been waiting hours and hours patiently. So what is the best reaction during my career? Others will have their pet reactions. Making this semisynthetic penicillin, bacampicillin. This is a prodrug. What does a prodrug mean? It means that this molecule isn't active. But what it entails to the molecule is high oral bioavailability. You will remember that many penicillins are quite unstable in the gastrointestinal tract, so you have to take them by injection. And this you don't want. This is orally available. So the pro comes in via this side chain. This is the pro part, which gives this opportunity. It was launched long time ago, and it became a very successful product in volume terms. We didn't make a lot of money because antibiotics, cheap products. Everyone expects antibiotics to be extremely cheap, and this one wasn't too expensive either. So this this stereo center that you create in the reaction that I will show you in a second is actually an epimer, and it isn't fully stereochemically defined. So it's a mixture of both epimers, one more than the other, about 55, 45, for some reason. This is the history of antibiotics. We go back to the days of Pasteur, contributions by Fleming, and then of Flory and Chain. Other antibiotics came to the scene, cephalosporins included. The Nobel Prize was awarded to these people, Fleming and Flory and Chain. You can joke about penicillins. They are so important. I asked our previous research director of AstraZeneca, what is the most important contribution in your eyes, in your lifetime, made by the pharma industry? Antibiotics. Before we had access to antibiotics, people died by simple infections. That doesn't happen anymore. The death rate in infections, at least the ones that we see in the Western world, is pretty low. There is but one downside with the penicillins, and that's the resistance. And this is something we need to grapple with. But this class of compounds that the penicillins produce is just phenomenal. We will have difficulties finding something that can do the trick as well as the penicillins did. So asking for a gift to someone who has everything, the obvious answer is penicillin, because it cures everything. I find that quite amusing. We're almost done. This is the first step in the process. You start from a commercial, very cheap, fermented penicillin G, benzyl penicillin as it's called. You react with this carbonate ester with a halogen here, and you get your intermediate, the ester. And we've done this in 3 ways. The chlorocarbonate first in acetone, high temperature, long reaction time. It was a black, I'm not exaggerating, a black reaction solution. But the yield that we could estimate was about 90%. So what's the big deal? 90% is good. Everyone would praise it. We worked with a Japanese company in those days. The Japanese are very clever. They had the license for Japan. Yoshitomi is the company's name. And they actually thought they could do something about this black, because the black color, as you know as chemists, always indicates that the chemistry has gone a bit too far, or we have some degradation and stuff like that. So they switched to DMSO. They could lower the temperature. 10 degrees doesn't sound as dramatic, but it gave a dark but not black product. And the yield went up to 97%. And this gave a better yield in the total process. But the breakthrough came when we all of a sudden got access to the bromo analog of diethyl carbonate. A very unstable or tricky compound to make, as I will show you in the next slide. Keep the temperature of the Japanese, but half the reaction time. This gives slightly yellowish, quantitative yield. I did the first experiments there. Was a bit uncontrolled of how I applied the temperature, etc. It just blew out of the reactor. It was so fast. I hadn't imagined that. I thought you would need to heat it a bit, but no way. How do you manufacture bromodiethyl carbonate? And here is some photochemistry at the end, not very commonly seen in the pharma industry. Start with diethyl carbonate, bromine, to get you to this product. It's the only feasible route. Trying to substitute the chloride with bromide is impossible because it resubstitutes again. So you only have to make it in a directed way like this. Overbromination is not what you want to see, so a flow process, this is in the mid-1980s, so unprecedented using flow chemistry here. Main impurities, dibrominated analogs. Optimizing showed that running it to 35% bromination degree was optimal. So, as this says, you charged diethyl carbonate, bromine, and you boiled it. It just was a very vigorous reaction under lighting, and up up to 35%, and then you distilled off the diethyl carbonate, you turn cycle, and then you purified the product, the bromodiethyl carbonate. And this high temperature also, because HBr is nothing you want to accumulate in your process, so boiling it at this temperature ensured that every bit of HBr just got blown off. You had to design a cascade reactor, 3 chambers, glass chambers. It was a spectacular process. We produce 100 tons of this material every year. And the collaborator from Switzerland, André Braun, he was really instrumental in making this happen, because our photochemistry expertise wasn't enough to really design this kind of process. So this brought us to the final process, which makes it so beautiful. So you bring in, this is the ester. No, let me see. Yes, this is your intermediate ester, and then in a in situ process, you have now to cleave off this side chain and replace it with phenylglycine instead of this benzyl group. PCl, and this is a method that the Dutch scientists came up with around 1970, so we didn't invent it, but we improved it and we trimmed it. PCl5, methanol, you go from the amide to the aminochloride, so you have a double bond between nitrogen and carbon and chlorine up here, and then you substitute the chlorine with a methoxy group, and then you hydrolyze it off, so you get the free 6-aminopenicillinic acid here, and then you throw in your new side chain and you have the product. This goes in one go. The yields when we started this work was around 30 to 35%. It was not a stable process. When we finished it, it was about 80. And calculating, making the math, if you make 100 tons of a material, that's quite a saver of material, energy, and cost. And so I'm extremely proud. This was a great experience to be involved with. This is just to summarize where I think we are going. Key technology concepts. You cannot deal with complex chemistry like I've described all day without having access to certain technologies. Green chemistry and sustainability is something you need to be aware of and to implement as much as ever possible. This is the paper. Bear with me. This is the paper of former colleagues of mine about solvent selection. If you're interested in how to select the best solvent, I recommend this. Others have done similar work. Move away from transition and precious metals. Go from batch to continuous. Intensify your processes. You can produce more from smaller volumes. Quality control, and you have objectives to fulfill. Maybe not all in one go, but all these kind of things you need to bring with you when you develop a process. CH activation is a question mark. You see a lot of fascinating papers, how you can selectively substitute hydrocarbon substituent. Maybe this can take away a lot of the chemistry we're used to today. Microwave has again appeared as a technology that might have a future. Chemistry, I don't need to convince you, is a core competency for making drugs. It needs to be involved in making the active molecule. The structures are complex and increasingly complex. Analytical chemistry, you need to know what you're doing. You need to understand how the chemistry works by taking samples, analyzing, etc., building the bigger picture. Environment, continuous manufacturing, and be open-minded to innovations, new science and technology. And we are living in a data-rich environment. Some call it big data, so much data generated. And how do we cope with this as chemists? How do we handle it? We have analytical tools that can record, well, thousands and thousands of data points. What do we do with them? We have computers who can give us a helping hand here. Self-optimization, that's a concept that some people pursue. We have algorithms are everywhere in society these days. They do all sorts of miracles for you. Maybe that is something you can exploit more. These are papers from people who are actually advocating moving to algorithms, data storage and retrieval. How do we handle management? How do we get access to data? And there is a visionary goal out there. Totally automated synthesis, flow chemistry, real-time analytics, these all present algorithms. So what these people who wrote these Science and Chem Society review papers advocate is maybe in the future, maybe in the near future even, you will be able to make molecules without human interference. The machines take over. Who knows? That's the end of a very, very, very long story. So I will only thank you for your patience. I hope I could deliver on my promises. To show you what process chemistry and what the pharmaceutical chemistry is about. My only words to you is having hard times. I know how tough it is here in Brazil. You should know that chemists per se, scientists, also are facing tough times in other countries. We need to stay sharp. We need to show the evidence, why we are needed, why we are important, how we contribute. Speak on behalf of your science. So reach out. Chemistry is, at the end of the day, much of a teamwork, and the team doesn't have to sit in the same building. The team can sit in different countries, all across. We are all united in solving major problems that we are facing. So I wish you good luck. If you have any questions, if you still have the energy to ask questions, I will be here. So, and I will be here tomorrow. You can save the questions till tomorrow. So thank you for your attention, and good luck in your career to become top chemist. Thank you.